you've learned about gametogenesis and the process of meiosis that forms those gametes, let's talk about genetic variation and problems that are associated with meiosis. The first thing we talked about was crossing over, and this happens during prophase 1 of meiosis. And what crossing over can potentially do is create more genetic variation, because during the pairing of the homologous chromosomes, which is called synapsis, part of one chromosome may be exchanged with a part of another chromosome. So if we look at this diagram here, we can see that this chromosome, which consists of two sister chromatids because they were replicated, this one came from the dad and this one came from the mom. Okay, they're homologous because they contain genes that code for the same traits. So you'd find genes maybe for hair color and eye color on both of these chromosomes. And it's the alleles that would have different codes so that we have the characteristics we do. So when these combine during synapsis or pairing during prophase 1, you can see that some of the chromosome may be exchanged with some of the other chromosome. So now the genes that were on this segment of the chromosome are now attached to this chromatid, and the genes that were on the other chromosome are attached to the other chromatid. So that will now give you a different assortment of genes. So that's going to create some variation. The next way that we get genetic variation is independent assortment. So there's two possibilities here. In red, we have the maternal set of chromosomes, and in blue is the paternal, so mom and dad. What happens is during meiosis, the chromosomes are separated into separate cells. So this would be meiosis 1, and then this is metaphase 2. You see the chromosomes are lined up. And then as the chromatids are pulled apart, we can get different combinations of chromosomes in the new cells. So here we end up getting all of the paternal chromosomes in these cells and all the maternal combinations in these cells. But over here, when they lined up in meiosis 1 or metaphase 1, they switch places. So now we can get some of the paternal chromosomes in one cell and the maternal chromosomes in the other. And if you notice, they're different sizes too. So here we get one of the big paternal and the small maternal, and here we get the big maternal and the small paternal. So it's just randomly assorting the chromosomes, which have different genes on them. So now if one of these were a sperm cell, it combines with a randomly combined egg cell, and we get genetic variation. So the two ways that we get genetic variation through meiosis are crossing over and independent assortment. Now let's take a look at how we can see some of these things and talk about the disorders that can happen during this process. This is a karyotype, which is basically a photograph of the chromosomes. This is human karyotype, and there's 23 pairs. These are homologous chromosomes. So we get one of these chromosomes from our mom and one of them from our dad. And they're mapped out into this, organized picture so that we can see kind of a synopsis of what's going on. The last pair of chromosomes, or pair 23, is always the um, sex chromosomes. That's going to determine your gender. So this is what we're going to look at for some of the disorders. To get a karyotype, first we need to get some of the genetic information from the developing fetus. So a process which is called amniocentesis may be performed. Now it is an invasive procedure because, as you can see, they're actually inserting a needle into the amniotic sac, which is exposing the fetus to different kinds of pathogens because it's no longer completely sealed. It will heal and it'll be okay, but anytime you have to be invasive, it's going to could cause complications. So they only do this if it's really necessary. If a woman has had past instances of problems with births, or in an older woman um, trying to have children, they may do this as well. So this is how they're going to collect the fluid around the fetus. This would be the amniotic fluid, hence amniocentesis. And then they're going to take a karyotype. And what they might find is that some of the chromosomes may be together still. And we'll look at that, a karyotype that shows us that in a minute. But the process that would occur by is non-disjunction. So disjunction is the normal coming apart of the chromatids or chromosomes. Non-disjunction is when they don't come apart. So if we look at this diagram here, we see that we have our chromosomes up here, the homologs, and these two stay together. So what happens is when they split, these two still go together and they go to one side, and so they end up becoming, in the one cell, and then the other cell is actually missing some of the chromosomes. As a result, when they divide again, it'll divide naturally so that you end up with half the number of chromosomes in each cell. But you can see that now we have one extra chromosome in these daughter cells because of this non-disjunction. And then over here, because there's extra chromosomes from the one cell, we're missing chromosomes in the other cells. If it were to happen later in the stage, okay, if it happens in meiosis 2 instead of meiosis 1, we can end up with, instead of having two cells from the one cell that are n plus 1, we have one cell which is n plus 1, and one cell which is n minus 1. So n again would be the haploid number plus an extra chromosome. This one would be minus a chromosome, so it's short one. And then we would end up over here, you end up with still two normal cells. So depending on when non-disjunction happens, if it happens in meiosis 1, you're going to end up with two n plus 1 cells and two n minus 1 cells. But if you have non-disjunction in meiosis 2, you'll get an n plus 1, an n minus 1, and then two regular haploid cells. So it's just a matter of when it happens. So the disorders that come about with that is monosomy is a possibility, which is 2n minus 1. So 2n is the normal chromosome number, and it's missing a chromosome. And these disorders include Klinefelter's disease, which actually looks like it would have an extra chromosome, but it has an extra X chromosome because the X's didn't separate during meiosis. And having an XY means the person is male, but in Klinefelter's disease, because of that extra X, they're not going to be as masculine. So they may not have the strength, or their testosterone may be a little lower, but they can carry out a regular life, and you may never know that they have Klinefelter's disease. The other one is Turner syndrome, and it makes it so it's XO, so there's no Y, so they're not masculine, but they're feminine. They're just not as feminine because they don't have the extra X chromosome and tend to be sterile. In the other one, we have trisomy, which is, so monosomy is minus 1. Okay, we're missing a chromosome. And trisomy, we actually have 3 of a chromosome. So we end up with 2n plus 1. And we'll look at that here. So if we look down here, and this is a karyotype of Down syndrome. And Down syndrome is characteristic of children having, sometimes facial features are recognizable, and they'll have more trouble learning how to walk and do normal everyday occurrences, and are more susceptible to some diseases. If we look at this karyotype, we look at chromosome 21. This is Down syndrome. It's called trisomy 21 because that's the chromosome. It would always be chromosome 21. There was non-disjunction here, so we ended up with two number 21s coming to this embryo and the extra. So one of them was maternal, one of them was paternal, or two of them were paternal and one was maternal. And then this could be identified through an amniocentesis, and it's also more likely in older women to have non-disjunction occur and have trisomy 21.